The effectiveness of screening blood products for one transmittable disease. News report. What is a blood transfusion? A blood transfusion is the addition of healthy blood cells taken from an individual into an unwell patient. The blood is made up of several components such as erythrocytes, leukocytes, plasma, and platelets. Blood transfusion procedure. There are different types of blood transfusions. The transfusion of erythrocytes is the addition of only red blood cells into a body that is deficient. The transfusion method is needed to increase the patient's erythrocyte levels and restore hemoglobin and iron back into the body. Plasma transfusions help replace the proteins that are deficient in the blood to allow coagulation to occur, which helps stop severe bleeding as the blood starts to clot. Platelet transfusions are needed to restore platelet count for cancer patients, as treatment methods such as chemotherapy and radiotherapy can reduce levels rapidly. Platelet transfusions allow to restore platelet counts to normal, whilst taking treatment and reduces the bleeding symptoms such as nosebleeds and bleeding gums. The doctor takes a sample of the patient's blood to check the blood group to make sure it's the same as the patient's. During the specific blood transfusion, the individual is sat on a chair and a needle is inserted into the vein in the arm, which is connected to a blood bag. The blood is slowly making its way through the tube into the vein, which is circulated around the body, which is seen in image 1, the procedure of the blood transfusion. What is blood screening? Blood screening is the procedure that is taken after blood is donated. Before patients receive samples for transfusion, potential donors are first screened with a list of questions relating to their health, such as medical history, sickness in the past month, or contracted any recent infections. This is done to determine if they are healthy and if they have diseases which can be transmitted by blood, such as HIV, hepatitis B and C, syphilis, and malaria, which is shown in image 2. The potential donor's blood is then tested to see if it matches the patient's blood and the Rh type to avoid rejection by the immune system. It is then tested using single assays to find whether they are non-reactive or reactive. If they are non-reactive, the blood donation can be used for clinical use, whereas if it is reactive, it is repeated again to find accurate results to be used or discarded. What is HIV and the effect of blood? Human immunodeficiency virus is also known as HIV. It attacks cells that help fight against infection and weakens the immune system, making an individual more susceptible to other infectious diseases. There are medications for the treatment of HIV. This is known as antiretroviral. This comes in tablet form and works slowly by stopping the virus replication in the body, allowing the immune system to repair itself. It can be taken with another medication to acquire the undetectable viral load. Cause of the infection is mainly sharing of bodily fluids such as seminal and vaginal fluid, blood, and breast milk. It can also be transmitted through sweat, urine, and saliva. Prevention of HIV is wearing condoms to prevent bodily fluid exchange and to keep up personal hygiene. HIV mutates and replicates at a rapid speed compared to the speed that lymphocytes can fully control infection and prevent the severe development of AIDS. HIV also replicates with the residual T cells, which have marker complexes, making it hard for the leukocytes to identify that it is a viral infection. HIV can also establish latent infection in T cells and can remain invisible to cytotoxic cells, and therefore can continue to replicate further and create new virions. In present day, these are the reasons why it is hard to create and find cures for HIV, but treatments can also help maintain and regulate HIV to the point where it can become undetectable. HIV can affect the body's immune system by destroying CD4 cells, which are T cells, and leukocytes are needed for the prevention and destruction of pathogens. Image 3 shows the shape and structure of human immunodeficiency virus, also known as HIV. How can blood be screened for HIV? HIV blood screening undergoes two different types of tests, which are the combination of antibodies and antigen testing. The immune response of the viral infection of HIV responds by producing antibodies to fight against the infection. The antigen testing detects the P24 antigen that is present in the protein coding of HIV cells. HIV spreads rapidly, so the levels of P24 can be easily detected through blood testing. Another way that blood can be screened is viral load or nucleic acid test. Viral load is testing which is used to predict how fast HIV will progress and how much damage the virus has already caused. It does this by finding HIV RNA that sequences and codes for replication. Once this is found, an enzyme is added to the produce to produce more copies of RNA, making it easier to measure the amount of HIV in the blood donation. The nucleic acid test is used alongside this, as it is used to identify small amounts of DNA and RNA in the sample test, which can be seen in image 4. Other testing methods can be used, such as ELISA, enzyme-linked immunosorbent assay, and PCR, which is polymerase chain reaction. This can be used to blood screen children for the detection of HIV. The effectiveness of blood screening HIV. The effectiveness of the combination antibodies and antigen testing is only effective 3 weeks after coming into contact with HIV. This only leaves a very short window to receive accurate results, considering HIV as an incubation period of 1 to 4 weeks. Viral load and nucleic acid testing is more accurate, as it detects small traces of RNA and DNA from HIV, especially with a small window to retrieve accurate results. However, this type of blood screening is used less, as it is modern and expensive. To increase effectiveness of blood screening HIV, both types of testing should be used alongside each other to get a more accurate result.